I want to now go into the GNF data, show you quite a different graph, a lot more complexity within the graph. But I hope what you've begun to see here is that the graph really represents the numbers and how numbers are related to each other and how different things here pan out. Okay. So do we want to do pig GI tract or do we want to do the mouse atlas? Should we try pig GI tract? It's a bit less complicated. Yeah, and we're an agricultural research institute after all. Let's do the pig GI tract. Okay. So drop that in again and say just check that it's hit the right numbers. Okay. And we'll say okay. And again, this is a filtered data set. I want to just, again, it's a bit more complicated than a time course, but not as complicated as a full atlas. So what this is actually is I think, how many different regions? Probably about 16 different regions of the pig. So we made this pig atlas some time ago. The full data set for that is within that folder. And basically this was a bit like the mouse atlas, but with pigs. So argument, pigs are a bit more like humans than mice are. And actually you can do things with pigs you can't do with mice. So basically we a whole bunch of us went into the house and slaughterhouse and pulled in lots of pig tissues. And I think it was something like 70 different regions in the full atlas. Okay, this is just only, I used to study the gastrointestinal tract, so I was quite interested in that. This is about 16 to 17 regions from the tongue through to the colon. Okay. So it's running the layout algorithm. Okay. So now you can see the graph is a little bit more complex. We have lots of structure there. So structure is good. So structure means that you have things which are grouped into some expressions. If you load up a graph and it just looks like a big hairball, you don't have much structure. Okay, that's the nature of the data. It's just what it is. Now obviously we don't know whether this is good structure or bad structure, but you can see there's quite a lot of complexity in that graph. And what have we got now? We have, so if you want to know something about your graph, this little question mark here says that there are 5,000 nodes and 200,000 edges. Okay, it tells you something about connectivity, how many, there's an average of 75 edges per node. And you can see there that it's all formed into lovely structure. So again, running the MCL, now routine to you all. We can run this on. Okay, so a lot more structure, a lot more things going on with this data set, a lot more kind of biological variation associated with this. And I'm conscious of time. I'm just going to quickly go through this and then go through some of the things you can do. So again, too many columns, too much information. Deselect all, select symbol, gene description, atlas profile. Okay. So GI clusters, call it the GI clusters instead. Cluster 1, you've got a description. Okay. So this is a kind of an idea of an annotated subset. Okay, so I've already taken this data. So when you've got this data here, then the first thing that you have is this big graph. You have to go through it, check what is interesting, check you believe it, check it's relevant, you know, what is this, is it biological, technical? You get the graph, you have to work it out. But ultimately what you're going to do, to really understand this data, you need to sort of take it a bit more slowly. So the quick answer to any given data set takes you 20 minutes. Is there structure? Does it make sense? Do I see things that I'm not familiar with? When you get to the point where you get to this level, what I've done with this cluster is I've added annotation to it. So it's cluster 1 of this data set, okay? It's the biggest cluster. So cluster 1 is always the biggest cluster of genes. That I've called it the ileum jejunum. So that's the ileum, that's the jejunum. It's full of immune genes. So just going down here, we get past the ones which are unannotated. Let's find some CD antigens. So CD19, CD22, CD40, CD72. So that's a B cell signature, if ever I've seen one. But there's also other things going on here, and you'll also see that very familiar signature that we looked at just now, which is the cell cycle signature. Okay? So what we've actually captured here with this biggest cluster is immune biology of the gut. So the distal gut, your jejunum and ileum are full of immune cells, pairs patches, plus actually a lot more distributed immune cells. And the other thing about that is they're proliferating like hell. Okay, so even though the rest of the gut, the epithelium of the gut, is actually proliferating, nothing is proliferating as crazily as the immune system. So what we've actually captured here is 2 signatures, one of which is actually a cell-specific signature associated with B cells and I think T cells, but also we've captured the cell cycle, which is something which is going on crazily. So what is going on in those 2 samples? We also noticed that only one of the jejunal samples is very strong. There's a lot of discrepancy between our 2 replicates here. Having selected this tissue, it's very hard to get 50 foot of pig gut and work out where you are. So it's possible that in the jejunal sample, or one of them, I collected ileum, or vice versa. Okay? So the replicates here are not great, but the overall signature is very nice. So this is a signature here, very strong in tongue, one of the esophagus samples and the lower esophagus. Again, you go through these and it's pure stratified epithelium. Okay, what is different about those tissues that makes them different from all the other tissues that I've disclosed? Well, they're all very full of stratified epithelium. And if I go down the list, I'm sure I'll find lots of keratins in there. Keratin 13, 15, 5, 71, 78, okay? Full of keratins. Next list. So similar but different. So this is mainly in the tongue. And actually in one of the esophageal samples. And if I go down here, you'll see myosin, 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 myosin. It's muscle, okay? So you can take this big data up and you just basically divide it up into chunks and you can go through it. So this bit only in the small intestine, okay? What's the small intestine got? Lots of, look at those are all your transporters. So all these are epithelial transporters that take your food and basically turn it into, internalize it. And likewise you can go through here. And so what you have to do with this data is, first of all, you get the clustering, and then it's worth sitting down and working out what you think any given cluster is. If you're doing this kind of relatively complex biology, the only way you can do that is to begin to take it out and do it. So in the last few minutes that we've got, I want to show you how you export things, how you begin to sort of take this data from a nice view to understanding it to doing something else with it.